Hi everyone, my name is Makrina Diakaki. I am a PhD student based in Wageningen University and Research, where I work in plant health. And today I would like to talk to you about the project that I am involved with that is called the role of seed microbiomes in seedling disease suppression. So basically everything that's going to follow will somehow have to do with seeds, their microbiomes, or diseases caused by plant pathogens. And to be precise, when I refer to seed microbiomes, I am talking about microorganisms that are living on and inside seeds, so epiphytes and endophytes. In this presentation, I would like to take you through the partnership behind this project, what is the motivation driving us to work with this topic, what are the research questions that we are interested in answering, how are we planning on doing that, and then eventually closing with some take-home messages. So the overall title for the project is Changing the System of Seed Health, an initiative of industry and research towards a paradigm shift. And this is a public-private partnership project, so it is partially funded by the Dutch government, but also involves stakeholders such as seed breeding companies, as well as universities and research institutes. And the project has two work packages. Work package one is basically my PhD, so understanding the role of the seed microbiome. And it's also what I will be talking about today. And work package two has to do with biological control on seeds. So there is an urgency with changing the system of seed health. But before getting there, because this is what is motivating us for working on this topic, let's see what this system looks like at the moment. So currently, the standard commercial seed sanitation procedure involves seeds being disinfected, and that is done to eliminate pathogens that are residing on the seeds, although of course, in this way, the seed microbiome is also being eliminated. And then after this disinfection, seeds are coated with phytosanitary products, and this is a protective treatment in order to basically then protect the seeds from pathogens found in the soil, in the place where they will be sown. And in this way, this protective treatment facilitates the development of healthy seedlings. However, especially over the last years, there is an increase in the number of phytosanitary products that are being banned in the EU, so banned from use as seed treatment products. And basically, given that if nothing changes in the current system of seed sanitation, then seeds will remain unprotected. And you can imagine that this means that there is also a lot of interest in exploring alternatives relating to this. And this brings us to the seed microbiome, which is a dynamic source of functional traits that is said to play a role in disease suppression. And understanding if the seed microbiome has anything to do with seed health could be a first step into achieving this paradigm shift. And it is also what we are working with. Our project also has the added innovation of being one of the few attempts of identifying key microbial seed components for disease suppression. And it is also, to our knowledge, the first systematic study to include a large-scale screening of multiple seed lots for disease suppression potential. Apart from this large-scale screening, we are also interested in addressing four main research questions. Firstly, are seeds with an intact microbiome more resilient to disease? Our hypothesis here is that seeds with a seed microbiome that has an innate ability to prevent or moderate infection are more likely to become more infected if they're challenged with the pathogen after having been disinfected. We are also interested in understanding if the same microbial DNA extraction method can be used to work with different seeds. So if that were to be the case, this would allow for comparative studies to be done between different crop species, and that to be done in a consistent way. Then we are also interested in characterizing the microbiome of a resilient seed. So basically exploring the taxonomical composition of seeds that have this disease suppression potential, as I called it before, and understanding which taxa are differentially abundant in those seeds in comparison to ones that do not show this suppressive potential. And we are interested specifically in looking at this for bacteria and fungi. And then lastly, do environmental conditions affect this potential? So would the seed microbiomes of different seedlots eventually become more similar to each other with regards to their disease suppressive potential if their mother plants were grown in the same conditions? And to answer these questions, we have already started working step by step, starting with choosing model crops and relevant pathogens to work with. Usually, projects like this focus on one or two crops and one or two pathogens, but in our case, we're actually starting with an inventory of possible combinations. Normally, there are some preliminary data available before the start of the project that can give a type of direction, but since in our case, our first step is the proof of concept, does the seed microbiome play a role in disease suppression? We are needing to start from a much broader point, hence also this larger selection. So the crops that are involved in the project are beetroot, onion, spinach, sweet pepper, coriander, red fescue, and perennial ryegrass. And these crops, each of them has already been challenged with one or two pathogens in tests that in phytopathology we refer to as bioassays. And each of these bioassays for each of those crops involved 40 different seedlots. And this is something that we have already completed. So this is what I wanted to talk about here. So these bioassays that have already been completed were done with the aim of determining phenotypic variation in seed resilience. So basically for each seedlot that was part of a bioassay, seeds were challenged with the same pathogen either when untreated or after having been treated with what I termed here microbiome reduction treatment. So this is some type of disinfection, such as immersing in hot water, for example, that eliminates part of the seed microbiome. We have now collected the data and analyzed it for this set of bioassays, and we are already seeing that indeed there are seedlots for which this is the case. So seedlots for which seeds that are treated with a microbiome reduction treatment show a higher percentage of infection. Our next step will be to now choose a smaller set of seedlots and focus on these ones, and from these to extract microbial DNA from the seeds. This is something that I have already started doing trials with, and I can say that extracting microbial DNA from seeds is harder than I thought, partially also because there is no single universally accepted method for doing this. So as you can imagine, this allows a lot of space for trial and error. The next step, once we will have microbial DNA extracted, will be to characterize the taxonomy and function of the seed microbiome of these resilient seeds. And we are planning on doing this using metabarcoding specifically for bacteria and fungi. Before ending the presentation, I would just like to highlight some take-home messages. First of all, that seeds do not exist in isolation, and any type of treatment on them also affects the seed microbiome. And then that the seed microbiome, as our preliminary data is showing, can play a role in seed health. This is also something that we're hoping to be publishing soon. Also linked to that, seed disinfection is not always beneficial. And lastly, changing the commercial seed sanitation system can secure seed health. And also possibly in this way, also secure food production in a sustainable way. So if you work with seeds but not microorganisms, I invite you to think about the seed microbiome during your next experiment, because maybe this is a direction that will give you some new insights. And if you do work on this field of research and you work either with plant microbiomes or seed microbiomes, I invite you to get in touch. Feel free to contact me and just introduce yourself and your research, especially since unfortunately we cannot meet in person. It would be really nice to connect. Lastly, I would like to thank my colleagues and collaborators for their contribution in this project so far. I would also like to thank the ISSS for allowing me to give this presentation today, and of course all of you for your attention.